Hereby, I open this academic ceremony in which Ms. Peters will defend her academic thesis, microRNA 26b plays a major regulatory role in cardiometabolic diseases. I welcome you, wish you lots of success together with your paronyms. I welcome also your supervisors, Professor Biesen, Professor Jankowski and Van der Forst, and also the members of the opposition, whom you will meet later, and of course, the audience here present and online. But before we start the opposition, I would like to invite you to present what you have done during your research period.
Thank you. Thanks to everybody for showing your support today. I'm very happy, proud, and very nervous to present to you a big chunk of my life. So 4 or 5 years of my life, I've worked on this topic. And many of you have asked me during that time, what did you actually do? And I never gave you the courtesy to explain to you in more than 1 minute what I actually was doing. So I hope today that I can present to you what I did for 4.5 years. So let's first go to the thesis title. microRNA 26b plays a major regulatory role in cardiometabolic diseases, which is a mouthful. So let's dissect the title. At first, what are cardiometabolic diseases and why would we need to study them? So cardio relates to heart, metabolic relates to metabolism. So when you take in food, your body will convert it into energy, which is metabolism. So cardiometabolic diseases are diseases like heart attack, stroke, diabetes, but also fatty liver disease. And in these diseases, lifestyle plays an important risk factor. So do you smoke? Do you drink? Do you eat unhealthy, like high fat, high sugar? Are you physically inactive? So when you live an unhealthy lifestyle, you have a higher risk of developing these kind of diseases. When we look at the numbers, in the US alone, we already see that 14% of the population has a form of cardiometabolic disorder. And these people, they miss working days, they miss, or they are hospitalized for multiple, they use medication, and this all piles up to a huge economic burden of $17.3 billion per year. Besides that, we do not really understand these diseases, and we have no cure for it yet. So how can we tackle this? Well, the title gave it away, but we were looking at microRNAs. And microRNAs are, again, a whole blurry concept. So what are microRNAs and why are we looking into them? So we often refer to DNA as a blueprint of life. And this metaphor I took, hopefully to explain to you what microRNAs are. So when you want to build a city, so you want to build your body, you need to have a blueprint. So this is the DNA. And then we want to build a single house. And so we take a note of it, which is then your messenger RNA, so mRNA. Next come the ribosomes. These are the construction workers. And they will build your house, which is a protein. So this is how we come from DNA to mRNA to protein. And microRNAs, they are basically your project manager. So they will oversee how many houses are built and at what time point we build these houses. So these microRNAs, they are, of course, then important people or molecules in this case. And this is why we want to look into them. So these could have a massive function in the body. And we look in particular into microRNA 26b. And we looked into 3 different diseases. So we looked into platelet function, so related to blood clotting. We looked into atherosclerosis, so the clogging of the arteries. And we looked into fatty liver disease. And in all 3 topics, the overall umbrella problem might be, for example, inflammation or fibrosis. And microRNA 26B is known to play a role in these processes. So this is why we looked into the microRNA. Then we made use of a mouse model, one mouse which was complete, so it had everything present, and then one mouse group where we deleted the microRNA from the body. And when you take out this project manager, what happens to the project? Basically, that's what we were looking at. So then we looked first into the platelet activity. We looked into these mice we looked into this microRNA because we know that the microRNA is one of the most present microRNAs in healthy human platelets. And we took the blood and we saw that these platelets were clumping together way more when the microRNA was not present. Then we made use of this mouse model where we labeled the platelets fluorescently, and then we damaged the arteries in the intestinal area, and then you can visualize it on the microscope. So indeed we see in the pictures you can see the fluorescence, which is your platelets, and what we saw is that these platelets are more active, or they're more reactive, so they are faster acting, and they create larger blood clots in the end. Because the lack of this microRNA resulted in more active platelets, we also wanted to look into atherosclerosis because there's also a relation between those diseases. And we also know that the microRNA is one of the most present microRNAs in human plaque tissue. This time we made use of the same mouse model, but we put them on the diet. And this diet was not to lose weight, it was to gain weight. So this was basically, we explain it as the McDonald's diet, so very unhealthy, but it's speeding up the disease progression. So when we deleted this microRNA from the entire body, we saw that there is much more atherosclerosis present in these mice. And we also saw that it was a more advanced stage of the disease. What you can also do is delete this microRNA from specific parts of the body. And in this case, we had this deletion everywhere in the body except for the blood. And then we also saw that this atherosclerosis was still more severe in these mice. Then we looked more into the vasculature. We took an artery of a mouse, we perfused it with immune cells that were fluorescently labeled, and what happens, they will stick to the vessel wall, which is exactly the first step in atherosclerosis. And because we saw more of the disease, we would also expect to see more sticking of the immune cells, which is exactly what we saw. Then we looked into the microRNA, or we deleted the microRNA only in immune cells, and this also resulted in more atherosclerosis. Then we took out these immune cells from their bodies, and we observed them on inflammatory levels, and we also saw that these were more, showing more inflammation. Then we came to more a therapeutic approach, so we took these immune cells again from the mouse, and we treated them with lipid nanoparticles. So these are the vesicles in which you can carry your microRNA. And we compared, we treated it with these microRNAs versus a control, an empty control. And we saw that this reduced the inflammation in the mouse immune cells. And then we saw also when we take it onto a human plaque tissue, so sometimes, or when patients go into surgery to remove the plaque, they take the plaque, donate it to science, and then here we actually used those plaques, added the microRNA on top, and we saw that this reduced inflammation as well. So this is also translatable to a human situation. And when we injected these mice with the microRNA, we saw that there was also a large reduction of plaque size. So this also suggests that it could be used more therapeutically. Next, because we saw such an effect on the immune cells in atherosclerosis, these same immune cells play a big role in fatty liver disease. So this is why we made the link to fatty liver disease. Plus, we know that this microRNA is involved in liver inflammation and liver fibrosis, so like more like scarring of the liver. And we again use these McDonald's mice. So, and we looked at the 3 key characteristics of fatty liver disease. We looked at fat, fibrosis, and inflammation. And in all 3 cases, we saw that there was a huge increase of disease phenotype again. So these livers were more fatty, they were more inflamed. Then we also again observed the mice that were injected with the microRNAs itself to restore the levels of the microRNA. And here we saw that there was a reduction of fat in the liver. So this also again suggests that it could be used therapeutically. So to conclude or to summarize, we saw that in platelets, when you delete the microRNA, the platelets will be more active and form bigger blood clots. There will be bigger plaques and more inflammation in atherosclerosis itself. And we can also already show that you can rescue the phenotype in a certain sense by adding this microRNA again to the mouse. And in fatty liver disease, we see that the livers are more fat, contain more fibrosis or more connective tissue, and show more inflammation. So overall, we saw a more severe disease phenotype in these mice. I would like to thank, so I had quite a journey over those 4 years. I started in Munich and then in IPAC. So I would like to thank those people, also people who came over from the Institute, IMCAR in Aachen, of course, and then Maastricht, KRM. And would I presented like I did it all by myself, but honestly, there were a lot of people involved, emotionally also, but also a lot of support from the technicians. Couldn't have done it without them. The supervision that I've had was great. It seems to be that number 3 is recurrent in my thesis, 3 topics, 3 institutes, 3 supervisors. And yes, this it was quite a journey, but thank you all for your support. And I hope you understood what I did now, finally, after those 5 years. I would like to give the word back to the prorector.
Thank you very much for a very clear presentation. We will now start the opposition, and the first opponent is also the chair of the assessment committee, that's Professor De Wint. He's professor in molecular cardiovascular biology in the Institute KRM in the University of Maastricht. Professor De Wint.
Thank you, Mr. Prorector. Dear graduate, congratulations also from my side on this festive day, where you can finally celebrate your accomplishments that was written in this very impressive thesis, very colorful. I loved it. And it shows really the results of hard work in the past few years. Of course, I also salute and congratulate the supervisory team, graduation team, Professor Biese, Professor Jankowski, and Dr. van der Vorst for making her journey possible. But certainly, I don't want to forget the family and friends, because a PhD thesis and a PhD journey, a graduation, is really not possible without the support of your loved ones. So thank you for supporting her. Well, dear candidate, I wasn't hired only to give you compliments. I was also hired to do, we have some work to do. Yes, and in order to start, I would like one of your paranyms to stand up and read proposition number 7 for me.
Studying microRNAs is like trying to follow a single strand in a spider's web, where each strand connects to many others. This complex network makes it difficult to assess how one strand affects the entire web.
Yes. Yeah, that feeds in perfectly to a figure that I found, figure 9, page 46, that really looks like a spider's web. Can you explain me what I'm looking at here? Chapter 3.
Chapter 3.
Page 46.
Ah.
Yeah, that very colorful spider web.
Highly esteemed opponent. Thank you for your compliments, and couldn't have said it better. Yeah, so this spider web is a kinome tree. So what we did here, we took the platelets of knockout mice and control mice, and we checked for the kinase activity. And we did this through a kinase array. So you have a chip with peptides, you add your sample on top, and then we can see which peptide is phosphorylated. So, and based on that, you can, with bioinformatics, find which kinases are more active. And in this tree, this is basically the groups of kinases, and it highlights which group of kinases or branch of kinases are more active in the knockout mice compared to the wild type.
That's a very good explanation. Thank you. I finally understand that figure now better. But what I'm puzzling with is, are we now looking at target genes? It's the supervisor, you call it a supervisor manager, a microRNA. Are we now looking at genes that are directly regulated by this microRNA or indirectly?
So that's a good question. So in this case, we are not looking at genes, we're looking at kinases, and we cannot, first of all, we cannot say if the kinase would be overexpressed or if the kinase is more active. So that's the first question we would need to ask. And second of all, we cannot tell whether indeed it would be direct or indirect interaction of the microRNA.
Okay, and to go a little bit deeper into this rabbit hole, yes, a microRNA, like you studied it, has, and you can see that beautifully on the picture that you provided on the cover of your thesis, it has this hairpin, and it has 2 strands, yes, and each strand, you know where I'm going to, each strand can have specific target genes. So now you studied a mouse that has a knockout of the complete hairpin. Are you not studying a double knockout?
That's a good question. I would say it's a complete knockout of the microRNA. The complete gene is lost. So I wouldn't call it a double knockout. I think it would be a good idea indeed to go into single strand deletion. So for example, if you want to go, for example, in vitro, you can look into the mimics and then only load your lipid nanoparticle with a 3'P strand and then a 5'P strand, and then also do the combination of 3'P, 5'P. So we can see what is the effect of the individual strand.
Yeah, so the 5'P or the 3'P strand.
Yeah,
yeah, beautiful, beautiful. And I mean, you make it very easy for me, but you already started about therapy. So you used LNPs, lipid nanoparticles, loaded with mimics. But since this microRNA is involved in so many phenotypes in different organs, how would a therapy look like? And most importantly, what side effects would you expect?
That's also a good point, and I agree with that we need to be careful with what we say. So I wouldn't immediately go and inject these lipid nanoparticles into a patient. So yes, you would need to have a lipid nanoparticle that specifically targets a cell type or a tissue via a specific receptor you could label it with, for example. So this is a way to directly go to where you want to go.
To the organ or the tissue or the cell type that you would like. But imagine we don't have time for that, and we want to tackle these difficult diseases, the cardiometabolic disease, and we just do an IV injection, so just in the bloodstream of this therapy. What organs might get show side effects? What would you expect?
I mean, the liver is clearing a lot of toxins in our body, so I would say that it goes through the liver, and I would say that here you would see the first effects, or your kidneys, for example. However, when we looked into these mice, it actually had a beneficial effect on the liver. We saw the decrease of fat. So in that scenario, I cannot say, give a definite statement on what side effects to expect, but I would say you would expect it first in the liver or the kidneys.
Very good. Also looking at the time and how many opponents we have, Mr. Prorector, I'm giving you back the word. I really try to make this candidate shake, but I cannot make her shake and get nervous. Thank you for the questions.
Thank you, Professor De Wind, for your opposition. The following opponent is also a member of the assessment committee. That's Professor Horneff, and he's professor in medical microbiology at the University Hospital in Aachen. And thank you for coming all the way from Aachen. The University of Maastricht is always glad to have guests from abroad.
Yeah, thank you. And thank you, dear candidate, for the presentation. I'm a microbiologist, so that means I'm quite far from the topic, and nevertheless, taking the thesis and your presentation, I think it's been becoming very clear what the question and the work was that you have been doing the last years. Thanks for this. I would like to maybe extend a bit on the discussion we previously just had on microRNAs, and you said project manager in your kind of cartoon-like kind of representation, and in your thesis title you say regulator, if you wish, regulatory role, a regulator. And I would like to challenge this term a little bit in the way microRNAs work. Could you allude on how microRNAs work? So you have this hairpin that was just talked about. What is the next step? What would happen to those molecules? What is between these hairpins and your phenotype?
Highly esteemed opponent, thank you for the compliments. I'm happy to answer or explain further. So I would refer to figure 1. And here we can see the synthesis of microRNAs. So here we can also clearly see that it will be processed within the nucleus, then it will be exported still as a hairpin, and then it will be cleaved into 2 separate strands. One of the strands will be then degraded, while the other strand will end up targeting the untranslatable region of the mRNA. And this is where the rule of thumb is that it would inhibit the expression.
Right, and you've been reading papers on microRNAs, I'm sure you did. And there are other ways of regulating, let's say, protein production. If you compare this, how would you rate this effect? And the way they act, and maybe what makes them different to, let's say, other ways of regulation of protein,
That's a good question. So I would say microRNAs, for example, they can be expressed over a certain time. So for example, this microRNA will be expressed during neuronal differentiation. And the interesting part is that its host gene is exactly the gene it will target. So over neuronal differentiation, the gene itself, the host gene, will become less, while the microRNA will become more. So I think it's more cohesive in that sense.
If you compare, for example, give me an example, a transcription factor expression or non-expression. gives you an effect on, let's say, protein expression in the end. If you compare this with the presence or absence of a microRNA, and you think about data that you've been seeing in papers, what is your impression?
So I do not have enough knowledge at the moment to answer your question in detail. I'm sorry.
Don't worry. I'm maybe a little bit provocative, but what I feel is maybe a little bit of a misconception for me. MicroRNAs tend to be more modulating factors rather than actually, in fact, regulating. So it's not 0, 1. It tends to be, put things a little higher or a little lower. On the one hand, on the other hand, they have their broad effect. So this is what I was alluding to. But let me shift gears a little bit and come to a different topic. And I think you've been using lots of different techniques and models, which I liked very much, and one in particular, and that because it's very, if you wish, very broad and in vivo, which was the bone marrow transplant as an approach. And I would like to, maybe you can totally explain again how this worked, what you did with the mice?
The bone marrow transplantation?
Right.
Yeah, so in this specific case, we used one group of knockout mice and one group of wild-type mice. We irradiated them, so they lose their bone marrow, so to say, and then we transplant the bone marrow of wild-type mice into these wild-type and knockout mice. So,
right.
Yeah.
And the sense of irradiation, what is the reason for this? Why are you doing this?
To destroy these hematopoietic cells there, so
right.
The bone marrow cells.
The bone marrow and recipient,
Yeah.
So basically you're replacing the bone marrow. That's
Yeah.
basically what you're doing. You've been doing this by taking bone marrow from this ApoE knockouts into either ApoE only or ApoE microRNA 26B double knockouts. There's in fact, there would be another combination that you could use to do this type of an experiment. So my question is, why didn't you do this? And which one would it be?
So we did it. So we did do the combination of where indeed you would transplant knockout bone marrow. Mm-hmm. However, this was outside of the scope of the thesis, so we didn't include it there. And to add on to your question is also when we look into these ex vivo perfusion experiments, there is also one group missing, which would be knockout leukocytes. And that reason why we didn't look into that was basically technical error. So this was the reason.
Right, but I think you can defend this, that you had a phenotype in immune cells, so I think it's perfectly logical. You have to design your experiment in a specific way. I think I agree with this, I'm just curious. And we alluded to the fact that you're using irradiation to remove the bone marrow. Is there an issue about this? How are you aware of problems that may arise at this point of the experiment?
So if I'm correct to what you're referring to, we do know that these mice, even if you put them on a 12-week Western type diet, they will still show smaller plaques. They're very fragile, these mice, so I think it does affect the phenotype in the end. I don't think it's fully comparable to the full body knockout on 12 weeks. We can also see it's much larger plaques, and this is just because they've been not irradiated.
Right, and you allude, and this is the conclusion you draw from this is a stromal effect, right? Which is interesting because you had effects on the immune compartment before. Mm-hmm. And now with this bone marrow experiment, you saw that there's something else, there's something on the stromal side. What I was talking about is that sometimes if you irradiate the bone marrow, there are certain cells that survive this. So there's an issue with this technique, and what's interesting is that this particular macrophages tend to do this, and they're part of your phenotype. That's why I was saying this. Okay, with this, thank you very much. I hand over back to the.
Thank you very much for your opposition, Professor Horneff. Then we come to the following opponent, who's online, and that is Professor Matic. She's also a member of the assessment committee, and she's professor in translational vascular medicine in the Karolinska Institute in Sweden. And thank you, Professor Matic, for coming all the way from Sweden, albeit online, of course. The floor is yours.
Thank you very much for inviting me. I am always very keen to be part of the thesis defense in Maastricht University. Right now I am in Crete, but happy to participate here. Dear candidate, I would like to congratulate you on a very broad and at the same time very deep thesis, because you managed in your 5 years of work to cover an amazing amount of molecular biology methods. And together with your supervising team, you also have managed to cover the most important aspects of cardiometabolic diseases. So I think from an educational perspective, you must have learned a lot in these 5 years. And so with these congratulations, I also would like to ask you a couple of questions. So from my own perspective and interest, I have worked a lot with human atherosclerosis, and my first question to you will be if you can maybe compare a little bit the early and the late stages of atherosclerosis as you see it in your mice, because you either feed them 4 weeks of high-fat diet or more, I think 8 or 12 weeks of high-fat diet, and how do these early versus late stages compare to the human atherosclerosis?
Dear esteemed opponent, thank you for the compliments. Thank you for the question. So here, the early stage of atherosclerosis. Sorry, can you rephrase the question? I'm not sure I got it.
Yeah, maybe I can ask in this way. So do late stages or advanced atherosclerotic plaques in mice, do they really replicate all typical features of late stage atherosclerotic plaques in humans?
No, they don't, because, for example, in this mouse model, the advanced stage, we don't have neovascularization in the plaque. We barely see plaque rupture. This is missing.
Yeah, exactly. Calcification, I guess, is missing as well, and so on. Exactly. And this is important because in order to assess the stage and the advancement of atherosclerotic plaques in mice, one also, and that is what you have done in your experiments, you have looked at specific features of these plaques. And so you came up with a conclusion that the plaques are very advanced in the knockout mice that you used. They were also very big. And at the same time, they had a lot of smooth muscle cells and a lot of collagen deposition. And so that is why I'm asking this question, because this is a little bit contradictory, one would say. So the question is, do you think that these plaques are more dangerous or not?
So that's a good question, yes. So we say that when there's more collagen, more small muscle cells present, these plaques are more stable and hence less dangerous. So based on that, I would say it's less dangerous. However, that's also maybe a too strong conclusion to draw, because we see a really massive difference between the wild type and knockout. It's not a very small percentage of difference. So I would still stick to the statement that it is a more severe disease phenotype.
Yes, I would agree with you. And this phenotype in human patients exists as well. These patients are operated, but they are recorded as being asymptomatic patients. So they usually will not get a rupture, but because the plaque is very big, they need to get operated anyway. So maybe just to go a little bit deeper on the mechanisms. So you have a lot of smooth muscle cells, a lot of collagen deposition. This is actually a similar phenotype as you see in the livers of these mice. And so that could be connected in a way in two different organs. And you have looked at the lymphocyte profile. And I'm wondering a little bit, have you looked specifically at Th17 cells? Do you know what is the status of those cells in your knockouts?
So the answer to this question is very short. We didn't look into that.
Okay.
Yeah.
Do you know if there is a connection between TGF-beta signaling, IL-6 signaling, and IL-17 signaling as well, and the effects of those cytokines on smooth muscle cells?
So I'm going to try to answer this question based on what I still know. I mean, the pro-inflammatory cytokine IL-6 will most likely result in a phenotype switch of the smooth muscle cell. Other than that, I cannot elaborate on this question.
Yeah, but here you are correct again. Indeed, IL-6 will lead to the pro-inflammatory, to the phenotypic switch in smooth muscle cells, but it also acts as an agent that will stimulate their proliferation. So it could serve to explain a part of the phenotype that you see, at least. And the similar goes for TGF-beta, and IL-17 also in this context has been shown, especially in atherosclerotic plaques. So I think this could be a suggestion for you, if any of these studies are continued, to maybe look specifically at the link with IL-17 and Th17 cells within plaques, and then the signaling that goes via TGF-beta and IL-6, because that could be the explanation perhaps for the phenotype that you see. And my very last question, because the studies that you did to test the therapeutics were so interesting, and so convincing, in fact. So the question is, when would you administer this as therapeutic agents? I think you hinted that it should be early in the development of atherosclerosis. How would that work?
Yeah, that's a good point. So I would first maybe look into another way of treating these mice. So you could, now it's more like a preventative approach, because we started injections at the same time as the Western type diet. So I would now maybe first do 4 weeks of Western type diet and then start with the injections to still see if we can still see effects on the phenotype. And then you will also be able to better answer the question, when we would deliver this therapy to patients.
Yeah. Excellent. Thank you very much. I'm very satisfied with your answers, and I'm happy to give the word back to the prorector.
Thank you very much, Professor Matic, for your position. The following opponent is also online, and she's also a member of the assessment committee, and that is Dr. Poteau. Her expertise is immunology at the French Institute of Health and Medical Research, and that is in Reims, and Reims is in France. And thank you for coming all the way from France, albeit online. The floor is yours, Dr. Poteau.
Thank you very much. Thank you, Mrs. Peters, for inviting me to your defense. This is a first for me also in your country, so I'm also a little bit nervous, so don't worry. I liked your proposition, your sentence proposition, and my question would probably refer to sentence number 3. About inflammation and leukocyte adhesion. So you very clearly demonstrated in your work that miR-26 controls adhesion of platelets, controls adhesion of monocytes. We also know from work of Mr. Weber, for instance, that leukocytes depend on platelet for adhesion to the vascular wall and plaque formation. So my question would be, do you think the effects you see about miR-26 on macrophage adhesion to the vascular wall would depend on the role of platelets and cooperation between platelets and leukocytes, which you did not you didn't test in your study, I think?
esteemed opponent, thank you for the compliments. Thank you for the question. We indeed, when we look at the leukocyte adhesion, actually, I think we could carefully state that it would be an endothelial cell effect, because if we look at the in vitro studies and their endothelial cells where we overexpress the microRNA, we see already these clear effects on adhesion. Plus, when we look at the ex vivo perfusion model, we also only look at the vasculature, so not the platelets, and here we also see a clear effect on the adhesion. However, this would indeed also be an interesting part to look at in the future. Yeah.
Thank you for your convincing answer. So in relation with that, my next question would be, since 26 is such important in vascular cells, first of all, do you think it's endothelial cells, or it could be also fibroblasts or smooth muscle cells? And how do you think vascular cells and miR-26 from vascular cells would modulate the fate of immune cells? So as you showed in the different experiments you made, there is a tendency or significant increase or decrease of monocytes or B cells in the blood, which you can always see, especially there is an increase of classical monocytes or non-classical monocytes, which I think is very interesting and could explain the phenotype you observe on atherosclerosis. So do you think miR-26 would have a direct effect on monocyte fate, meaning progenitors of monocyte of B cells, survival of these cells, apoptosis of these cells? Would it be a direct effect or an indirect effect of miR-26 from the vascular cells? I don't know if my question is clear.
So if I understood correctly, it's two questions. So the first question was whether I would only expect endothelial cells to be the cause. I do not, I wouldn't think so, because, I mean, as you stated, there are also smooth muscle cells and fibroblasts present. So this would definitely be something to first look at, either with a specific knockout model or to be more animal friendly, use the lipid nanoparticles in vitro. And the second part of your question is whether the vascular MIR affects the fate of the leukocytes. That was your question? Yeah.
Yes, exactly. Because you have a less or more monocyte and B cell in circulation. So this is a direct or indirect role of miR-26 absence or presence.
So I would say that it is probably an indirect effect, because if you have so many cell types involved, especially if you look at the full body knockout, we already see these effects on the B cells, as you stated, and also we see a decrease in leukocytes. So I do think that the microRNA, you can say it's involved directly in, indeed, the leukocyte formation or homing, or it's the effect of the endothelial cells with the adhesion and transmigration.
Okay. Yeah, what I meant is that maybe you answer right. What I meant is probably either miR-26 could be encapsulated in microvesicles liberated from the vascular wall and transferred to the immune cells and maybe act directly on immune cells. Or it could modulate cytokine production or growth factor from the vascular cells that could act on hematopoiesis, for instance, or homing of cells. And my last question, because you like the number 3, so I had 3 questions. A very easy question. So I saw you published 1 paper already on platelets. I don't think the other 2 stories are published yet. Are they under publication? And if not, would you like to compile the 2 stories or make maybe 2 papers out of the 2 studies?
So actually, yes, I would make 2 more publications out of it. Actually, we're making 3 out of it. So we have the myeloid microRNA side, we have the vasculature, and then we have fatty liver disease or MASH. So these would be 3 papers then.
Okay, thank you very much, and congratulations for your presentation and your manuscripts.
Thank you, Dr. Poteau, for your opposition. We come now to the following opponent, that's Professor Winkels. And he's professor in experimental cardiology of aging, and that is in the University Hospital of Cologne, and that is in Germany. And thank you for coming all the way from Germany.
Yeah, thank you very much for inviting me. It's a great pleasure here, and I want to start by congratulating you, dear candidate, on this very elaborate tour de force on unraveling microRNA 26 in cardiometabolic disease. Also extending these congratulations to your promoters and supervisors. And as my job is today also to ask you a few questions, I want to circle back to something that has been discussed before, and that's the potential use of these LNPs loaded with a mimic to restore the expression. And you already highlighted that they profoundly reduce atherosclerosis, also have a benefit on the liver, yet I noticed that there is a massive increase in cholesterol and triglycerides in the circulation. So how would you integrate all of that, as cholesterol and heightened triglycerides are risk factors for cardiovascular disease?
Highly esteemed opponent, thank you. That's very well noticed. So first, of all, regarding the increase of cholesterol and triglycerides, the microRNA does have an effect on the uptake receptors, on SRA, and I think this would be the explanation why we see more cholesterol and triglycerides in the blood. This is, of course, not beneficial to atherosclerosis or heart diseases or any diseases, so this is definitely I would say a drawback from the use of these lipid nanoparticles.
Yeah, thank you. This makes a lot of sense. Yeah. So, and as you already pointed out, these particles have to be seen with care if translating them. And something that I noticed throughout your studies is that you're focusing very much on the aortic arch and also on the thoracic, sometimes abdominal aorta, which is, of course, a major site of atherosclerosis development. But I noticed here that in the aortic root, you barely see differences in many of the models that you have been using. Do you have an opinion or an idea why that might be?
That's, again, a good observation. So what we know is that the aortic root has a different embryonic origin. So this is one point. And the second point is shear stress. So in the aortic arch, there's shear stress where which we can also see, for example, or it's supported by my in vitro work with the endothelial cells. So these endothelial cells are more prone to inflammation. It would be interesting maybe to do an in vitro experiment where we put shear stress on the endothelial cells and see if this hypothesis is correct.
Thank you. Yeah, I agree. And of course, you have, for example, PVAT that is more pronounced around the aortic tree than within the root. Also, there's a little bit of fat. But that brings me to the question, if you can allude to where is actually microRNA 26 expressed. We have already talked about monocytes, platelets, endothelial cells seem to play a role. You have mentioned neurological development. So where do we see this microRNA?
So this microRNA is globally expressed. So I think you're aiming for adipocytes. So I guess we did also try to start up a project there, but that was outside of my PhD topic, but it's also expressed in adipocytes, for example.
Okay, great. So yeah, that makes sense. If it's globally expressed, it would be likely also playing a role there. And maybe as a last question, I want to shift a little bit gears and pick up what Dr. Poteau has been asking about a potential role for development of immune cells. And I noticed in your myeloid-specific deletion that there's actually one cell type that shows a profound difference. In your macrophage phenotype seem to be rather mild, but you have a lot of neutrophils in your circulation in relativity to the B cells. So why do you think that might be? And also pointing a little bit towards the model of the LysM to help you there. So what do you target with the LysM?
So with the LysM, you target your myeloid cells. So we would delete specifically the microRNA in these myeloid cell lineage. So that includes neutrophils, macrophages, monocytes, megakaryocytes, so we also look at the platelets. And could you rephrase the other part of your question?
No, thank you, exactly.
Okay.
So what I wonder is this effect even more driven by the representation of neutrophils, and does this deletion in neutrophils actually drive your phenotype rather than the deletion of microRNA 26 in the monocytes, as your phenotype in the monocytes and also the BMDM seem to be rather mild, yet you have this profound difference? What is your speculation there?
So this is a good point, a good suggestion. We did not look specifically into neutrophils, so I cannot make a definite statement on it. But we would need to look maybe in a neutrophil-specific knockout or neutrophils in vitro to see what the role is. I would actually think that neutrophils do play a role as well. I mean, they're important drivers in atherosclerosis, so I cannot imagine they don't have a role here.
Yeah, thank you very much. Thank you for addressing all my questions, and I'm handing back to the prorector. Thank you.
Thank you very much for your opposition, Professor Winkels. We now come to the following opponent, and that's Dr. Kremers. His expertise is inflammation, and he works at the Maastricht University Medical Center. And today he has also accepted the role of secretary of this committee, for which I am very grateful.
Thank you. So, first of all, dear candidate, congratulations with your thesis. Also to the supervisors and the family and friends. It's a really nice thesis, a nice cover, and it was a pleasure reading. However, I still have some questions, so I hope that you can answer them. First of all, starting with a simple question. In which other diseases do you expect that microRNA 26B can play a role, or can potentially play a role? Yeah.
Esteemed opponent, thank you for your compliments, thank you for the question. Since it plays a major role in inflammation, as we've seen, and also in fibrosis, the first disease that I would link to this would be, for example, chronic kidney disease. Could also look at other inflammatory diseases like rheumatoid arthritis. I would say any of lipid, inflammation, or fibrotic related diseases. That is what I would further look into.
Okay, very good answer. Yeah, indeed, all kinds of inflammatory diseases will be affected. So potentially there'll also be a role as a treatment for it. Can you elaborate a little bit on that? How do you see microRNA 26B to be a therapeutic or biomarker, or how can it help the human health mainly?
That's a good point. So actually, I would first vouch for this microRNA to be more used as a biomarker instead of a therapy. If we look at the, for example, the last figure of my thesis, so that is in chapter 3, figure 32, and we see that in liver cirrhosis patients, these microRNA both strands are massively overexpressed compared to healthy patients. And this is taken from the plasma, so it's less invasive than, for example, taking a liver biopsy, which is now the golden standard. So taking the blood from a patient and then the plasma and measuring these microRNAs could give us an indication of what stage of disease they're in, for example. Also, I know microRNAs can be used as a prognostic marker. How is somebody going to react to a therapy, which is also very interesting. And therapeutically, I think, as we already highlighted with the previous discussions, I think there are still some limitations in that sense.
Okay, yeah, a clear answer, so thank you. So you mentioned in your discussion that microRNAs are highly conserved between species. However, you find some contradicting data between human and your mice models. So in human patients with atherosclerosis and MASH, you saw that the expression level of microRNA 26B is increased, while in your knockout, having not this microRNA 26B, you actually find the opposite. How can you explain this?
So we see indeed higher expression in human plaque tissues compared to healthy vasculature, and this could, for example, be a compensation mechanism from the body that it tries to rescue the phenotype. And same with the liver cirrhosis patient. So the overcompensation of the body of producing microRNA 26B might not be enough to press down the disease development. But maybe when you add it to it, it might be enough.
Okay, yeah. In addition to that point, so you loaded LNPs with mimic microRNA 26B to restore the levels, but how sure are you that you not overexpressed this level? Because also in light of potential therapy, you want to be at a certain level of expression.
Mm-hmm.
So
what we did is basically we anyways always genotype the mice whether they are really a knockout or not. So we know the expression of the microRNA in that sense. And then to check whether we come back to baseline, to wild type, so to say, we again check the expression levels of the microRNA in tissues. And you can, for example, also see where the microRNA will end up. So we fluorescently labeled the lipid nanoparticles. We then visualized that they actually ended up in the plaque, which was pretty nice. And it also ended up in the liver, of course. But it was good to see that it actually ends up in a plaque, inside a plaque, possibly via macrophages, or most likely via the macrophages, I would say.
Okay, thank you for the answer. So I think I've time for one final question.
Short question.
So I show that the LNPs you will target the right cells because it's also cell specific, of course, how this responds if you inject them.
Can you rephrase your question?
Yeah, so if you inject the LNPs, the lipid nanoparticles, there's a cell-specific response, right? So it will end up in a specific cell. How sure are you that the expression of microRNA 26B will end up in the right cells, which is the normal expression in wild-type animals, for example?
Yeah. So in that sense, I would say that Again, you would need to have, for example, targeting via a specific receptor. So you label your lipid nanoparticle with the receptor.
You may finish your answer. You may also say enough is enough.
It's enough.
Thank you, Dr. Kremers, for your opposition. Dear candidate, Ms. Peters, the time appointed for the defense of your thesis has passed, and the degree committee will now withdraw to discuss the quality of your thesis and the way you defended it. And I request that you and your company on site and online await the results of our deliberation and our return in this room. And I suspend the meeting.
Please all rise.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Make the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. To the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
I reopen this academic ceremony. Dear candidate, Ms. Peters, the degree committee here present has assessed the quality of your thesis and of your defense. And in view of its positive verdict and in taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. And Professor Biese is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Belooft u dat u altijd volgens de beginselen van wetenschappelijke integriteit te werk zult gaan? Eerlijk en zorgvuldig, transparant, onafhankelijk en verantwoordelijk.
Dat beloof ik.
Dat is de enige correcte antwoord. Krachtens de bevoegdheid, ons door de wet toegekend, volgens het besluit van de commissie hier tegenwoordig, verklaar ik hierbij u, Linsey Jeannette Francine Peters, tot dokter te bevorderen en u alle rechten te verlenen die daaraan volgens wet en gewoonte zijn verbonden. Ten bewijze hiervan overhandig ik nu de bul, door rector, secretaris en overige leden van de promotiecommissie ondertekend en met het grootzegel van de universiteit bevestigd.
Je mag hem helemaal opklappen. Voor de... Mag dichterbij komen. Als je hem even openklapt helemaal, dan kan de fotograaf wat dichterbij komen. Zo ja. Ja, dan moet zij vasthouden. Hè?
Ja, ik leer het nooit.
Moet de dokter dat niet vasthouden?
Oh ja.
Fotograaf, het is veranderd.
Nog een beetje. Ja.
Pictures we do now.
Ja.
Thank you.
Please be seated.
Ja, beste dokter Peters, beste Linsey. Het is een eer om deze gelegenheid vandaag te mogen afsluiten en om je als een van de eerste te feliciteren met je dokterstitel. Als je supervisor heb ik je onderzoeksreis van dichtbij gevolgd. En we zijn begonnen met een heel ambitieus idee om microRNA 26b even te onderzoeken. En door heel veel toewijding, hard werk, heb je dit omgevormd tot het zojuist verdeelde proefschrift. At this point, I would also like to thank all the members of the assessment committee on site and online for judging the thesis, and especially also for today for the lively discussions. So thank you very much. En je proefschrift en zeker ook je verdediging vandaag, en daar was iedereen het heel erg over eens, is een prestatie die niet alleen getuigt van academische excellentie, maar ook van persoonlijke groei, doorzettingsvermogen. En laten we beginnen bij het begin. Zoals je al zei in je presentatie, het begon allemaal in München, waar je als masterstudent bij mij in de groep kwam, bij Christian Weber en Yvonne Deuring. En deze ervaring vormde denk ik al een cruciale basis en droeg bij aan het fundament van je hele promotietraject. Gelukkig heb je naast hard werken ook tijd gevonden om te genieten van de lokale cultuur. Waaronder natuurlijk de iconische bierfeesten op het Oktoberfest. En heb je ook goede vrienden gemaakt waarmee je ook tot nu toe nog steeds contact onderhoudt. Het tweede deel vond dan in plaats bij Imker in Aken, bij Joachim, in nauwe samenwerking met Erik in Maastricht. En ik denk dat onder de verhuizing van het hele lab destijds en door eens coronarestricties vlak daarna, heb je project eigenlijk de hele tijd draaiende gehouden. Wat denk ik een erg knappe prestatie is. En de samenwerking met andere onderzoekers in Aken en Maastricht heeft je dan ook in staat gesteld om je onderzoek verder uit te breiden en verder te verdiepen. En je bent tijdens je onderzoek in aanraking gekomen met heel veel verschillende onderzoeksgebieden, van bloedplaatjes tot nanopartikels. En ik denk dat deze diversiteit in je onderzoeksfocus getuigt ook van je vermogen om nieuwe wetenschappelijke vragen te verkennen en ook een multidisciplinair onderzoek te omarmen. Een ander belangrijk aspect van je werk is je rol als mentor geweest. Met veel toewijding en deskundigheid heb je 3 masterstudenten begeleid. Cillian, Rosie, Leonida. En ik denk de manier waarop je deze studenten hebt ondersteund en begeleid getuigt ook van je grote betrokkenheid voor de ontwikkeling van jong talent en ook je vermogen om anderen te kunnen inspireren. Tot nu toe heeft je harde werk al geresulteerd in 3 reviews en een origineel artikel. En zoals je zelf ook al zei, er zitten nog veel meer artikelen in de pijplijn gebaseerd op je proefschrift. Daarnaast wil ik ook een moment natuurlijk nemen om de steun van je familie, vrienden, collega's te erkennen. Ik denk hun aanmoediging, geduld hebben ongetwijfeld een belangrijke rol gespeeld in het succes van je promotietraject. En vandaag markeert dan niet alleen het einde van een promotietraject, maar ook het begin van een nieuw hoofdstuk. Al is hij natuurlijk al een tijdje geleden begonnen bij Elmezitran. En ik ben overtuigd dat met de kennis en de ervaring die je hebt opgedaan tijdens deze promotie, dat dit een stevige basis vormt voor alle toekomstige successen. Dan ook namens het gehele promotieteam wil ik je dan ook van harte feliciteren met deze presentatie. En gefeliciteerd en veel succes.
Thank you very much, Dr. van der Forst, for a very nice laudation. Dear Dr. Peters, also on behalf of the Maastricht University and its deans, I congratulate you with the degree you have acquired, and I hope you can celebrate it, of course, and a big step in your career, perhaps one of the most important days in your career. And I also congratulate Professor Biesen, Professor Jankowski, and Dr. van der Forst, your supervisors, and of course also all your friends, family and colleagues here on site and online. I would like to thank the opponents for their work, and of course the technical support team and the PEDEL, because without their presence and help this academic ceremony would not have been possible. Having said that, I'm about to close the meeting. I have an announcement, and I'll probably do it in Dutch. That's easier for most of you, I suppose. Wij gaan hier nog een foto maken met de buitenlandse gasten online. Dus ik verzoek het publiek vast naar de refter te gaan. Dat is de gang uit en dan naar rechts. Daar is de receptieruimte. Wij gaan dan nog een foto maken aan de voet van de trap met degenen die hier wel zijn. Deze groep. De fotograaf gaat natuurlijk wel gezellig met ons mee. En als u dan bij die refter staat in die receptieruimte en u drinkt een wijntje of een kop thee of koffie en u denkt, wanneer mogen wij dan de dokter feliciteren? Dat is vrij simpel. U houdt ons in de gaten. Wij gaan eerst en dan komt u. Having said that, I close this meeting.